All right, how's everybody doing this morning? Yeah, I felt like it was a day for my heterozygote shirt. Huh? My wife found it. I think she found it on Amazon. She surprised me with it. I was like, oh, that's awesome. No happiness in the morning. Must be somber. Like a funeral dirge when you come in. All right, give you guys a couple of more minutes for people to straggle in here. Um, while I'm waiting on that, I'll say, so today's lecture, we have some more new material. Um, and when I say new, probably new to you, period, not stuff that you've had before and we're covering new. So by the end of the lecture, um, I'm going to show you some things that I just want you to get the general concept about, not necessarily all the deeper mechanics. At the beginning of the lecture, I'm going to talk about things that I want you to know the mechanics of for the exam. So, and I'll try to remember to make that clear. Excuse me, as I go through the slides. You would don't have anything to tell me the time. All right. I just have to trust the clock up there. Says you guys got 10 more seconds before I have to blather on. All right. So we've been talking about, let's see, did I? Yeah. The replication machine. And we talked about prokaryotic replication as well as eukaryotic replication. And we started to do some detail on eukaryotic uh, replication of DNA. You should be able to compare and contrast those two systems. You should be able to compare and contrast them, be able to talk about them. The differences, what's similar, what's the same. I mean, what's similar, what's the same. What's similar, what's different, yada, yada, yada. We talked about strand-directed mismatch repair. This is an uber important part of DNA replication. This machinery follows the replisome, the replication complex. And it also, we will see that there are other base pair exclusion repair processes used in other parts. You should know the functions of mut S and mut L and the general way of how they work and how the sliding clamp orientation determines which strand they're going to excise bases from, which is the new strand. They don't excise from the old strand. That's one of the really cool things. It's not just random. They actually excise the new strand. I mean, new strand. I'll get it right here. Boy. All right. We talked about topoisomerase 1. You should be familiar with this mechanism, including the tyrosine. That the tyrosine attacks that ester bond, one of the ester bonds of the phosphodiester bond, but that the energy is stored in that phospho bond to the tyrosine. So by the end of when it's all said and done, the stress is relieved. I cannot talk this morning. My brain's, I think, working, but my mouth isn't. All right. Anyway, once the stress is relieved, it religates together using the energy that's stored in the bond, and Bob's your uncle. Everything's cool. You should know about the general idea of how topoisomerase 2 works. Topoisomerase 2 is not as deep as in the mechanism about who bonds to who, but you should know how it's making a double-stranded break, hanging on to those two ends and opening up so that the other piece of DNA can slide through and then religating the double-stranded break, and that it does take ATP. All right. So we talked a little bit about the idea of fork initiation at the replication bubble. We talked about RNA primer synthesis. So, of course, you should know that in prokaryotes that we have DNA polymerase 1 that replaces the primers, and DNA polymerase III does all of the extension on both strands. On eukaryotes, DNA polymerase alpha is an RNA primase. DNA polymerase delta does the lagging strand and actually starts both strands off the primer. It's the one that starts off the primers. On the leading strand, it is quickly replaced by DNA polymerase epsilon. You should know these and be able to talk about these things. All right. You should know the general idea of the initiator proteins and how the helicases are actually pointing toward each other and have to pass each other as they start replication. And that the initiator proteins work on an AT-rich region of the ori to help destabilize it and get it pulled apart so that the helicases can start doing their job. All right. That's pretty much what we've talked about. The cell cycle I'm not going to talk a lot about because you should know that by now. If you don't, review it. On the initiation replication, yeah, you do need to know what's going on here. The MCM helicase, CDT1, CDC6. It would be nice if you remember those names and could talk about them. But if I ask a question about this, I'm willing, if you can describe what's going on and you don't get the names right, that's not going to bother me quite so much. But you should be able to talk about this process, including when the protein CD45 and the GINS complex comes in, the phosphorylation of the CMG helicases, the phosphorylation of the ori recognition complex, and the fact that we get polymerase epsilon up here and polymerase, why is that so epsilon? It should be delta. All right, there we are. There's delta. I don't know why they put epsilon up there. Just noticed that. Well, alpha and E loaded. Okay, whatever. Leading strand synthesis taken over by DNA polymerase epsilon. Yes, that is the one that takes over. All right. So, CMG, by the way, is CDC45 MCM GINS complex. That's how we get the CMG back here. Where did CMG go? Right there. The CMG helicases that require proteins to be activated. All right. So now I think we are going to start some new material. So as the helicases start unwinding the DNA, there is another impediment to replication, and that's the nucleosomes. Now, prokaryotes don't have this problem. Maybe they do. They do have some histone-like proteins, but we're not going to talk about that. So you have the DNA wrapped around the nucleosomes, and so you got to get the DNA off the nucleosomes because you need to get access to all of the DNA. You need to replicate the DNA. So right here in front of the replication machinery is this FACT complex. And the FACT complex facilitates the disassembly of the nucleosomes. But it's really, really kind of cool. Of course, everything I think is cool, so you take that with what it's worth. So our replication machinery is going this way. We need to get these nucleosomes off from the DNA, or the DNA off from the nucleosomes, however you want to look at it. This FACT complex comes along and it helps remove the H2A and H2B dimers, but H3 and H4 stay together as tetramers. Remember, the whole thing is an octamer. So H2A and H2B go off as parts. And what happens is, remember, we have the histone code, right? You have parts of the DNA you want silenced, parts that you don't want silenced. You don't want to start from scratch. So these H2, H3, H4 tetramers randomly assort between both strands. And then a new tetramer fills in on those, and then our H2A H2B dimers that was displaced come in and finish making our nucleosome. And we have this loading protein. So you should know that there are proteins obviously involved besides the FACT complex. The FACT complex is, when you ever hear the word complex, we're talking about a multi-protein, multi-subunit machine. All right. So that means that the histone code is still on these things. It doesn't disassemble the histone code. And H3 and H4 tetramers are randomly distributed. So as this comes back together, my reader-writer proteins can come along and go, oh, this is supposed to be heterochromatin. And then I'll start writing that on the ones that don't have the right code on them. Or this is supposed to be euchromatin. I'll start making sure all these guys are euchromatin. So that's how you can inherit the genetic code, not the histone code that controls the genetic code. That's epigenetic inheritance, because the histone's going to silence or make available that, and that process itself is heritable. Yes? Yeah. Yes, so when the FACT takes this apart, the H2A and H2B come off as dimers. The H3, H4s come off as, they stay together as tetramers, and then they distribute the parent ones between both strands. And then newly synthesized H3, H4s come in to fill in where they need to fill in. So the parent ones have the histone code for whatever it was for that section of protein. They hang out right there at the replication complex. I'm sorry?
Parent and synthesized.
Yes.
And H2A and H2B. When H2A and H2B displace from one octamer, do they sometimes mismatch, or do they all form back together?
Well, they're gonna have the histone code in that area as well, right? So they probably are going to randomly assort, but the big thing is the H3 and H4. There's going to be some these displaced H2A, H2Bs, H2A, H2Bs. We have to have new ones of those too. We've got twice as much DNA. We need twice as many nucleosomes. But really, it seems like H3, H4 is the key factor to the epigenetic inheritance. All right, cool stuff. I think that's really cool. You're like, hey, go to sleep. Okay. Now I got this this illustration. We're going to jump from the replication complex and the FACT complex and nucleosome disassembly and epigenetics to the end replication problem, the telomeres. We have linear chromosomes. Now, bacteria don't have this issue. They have one circular chromosome and extrachromosomal DNA in the form of plasmids, which are also circular and have their own ori. But we have the end replication problem. There are two problems. One is, we're going to talk about this 5' 3' overhang here. Actually, the 3' end is a bit of an overhang, and how that occurs. And we also have the problem that if we just left it alone, if we just finished copying the chromosome and said we were done, we have DNA repair complexes that look for double-stranded breaks. And it looks like a double-stranded break. So those repair complexes would start putting our chromosomes together, making them bigger chromosomes. So that's the two problems we have to deal with at the telomeres. Now, you're going to see that I put up a different sequence here. The idea is we have this telomerase machine. And part of that machine is an RNA template. And part of that machine is a reverse transcriptase. It's going to use the RNA template that's associated with it as a template to extend the telomeres. That makes sense? All right, so let's look at this from your book. This is the typical telomere sequence, but there are variants. There's not like, this is the sequence. So I'm not worrying that you know the sequence of the bases that the telomere gets extended by. You have this complex. We have some additional proteins here, but we want to focus on this guy right here, the catalytic core of the telomerase. As I said, it's made out of two things. It's made out of H-TERT. The H is for human. T for telomerase, and reverse, or TE for telomerase, and RT for reverse transcriptase. So the catalytic part of this is a reverse transcriptase. It's going to read RNA and manufacture or make complementary base pairing with DNA. Going to go the opposite direction that we talked about when we talked about the central dogma. But to do that, it has to have this RNA core. So all of this blue here with all these little loops and things, this is an RNA. And RNA can form complex three-dimensional structures, and that's what you're seeing here. And so we call that part of this whole thing H-TERF, for human telomerase RNA. So the sequence that gets added on is dependent by this guy, and it has to have a complementary part here to the telomere so it can match up, and then we can start extending things. All right. So you need to know about H-TERT and H-TERF. Obviously, other animals have TERTs and TERFs, but we're going to talk about the human one, so we're going to know human. The lowercase h just means human. All right. So the RNA both acts as a scaffold for additional proteins and for the reverse transcriptase catalytic core to make this machine. So what's going on with this machine? All right. How do I want to do this? I got to think about this. Actually, let's do it like this. So what happens? On the 3' strand, we're going this direction with a replication fork. It's our leading strand, and the complex can just run off till the very end. So this is our lagging strand. So we need a primer. So we're going to put our primer where? There's no place to put a primer. Once I get to this point, I can't put a primer where there's nothing. I can't replicate that. I can't build without a free 3' hydroxyl. This is my 5' end. I have to build this guy going this way. I can't put a primer there. Now there's been some current research. We actually think it's more complex than this, and there's some 3' stuff going on. We're just going to go with the classic mechanism right now. So you have this end replication problem. You're going to wind up with these tails, these 3' tails, and shorter 5' sections. And if we go back to this guy, there's my 3' tail, this being extended, and my 5' guy, which is actually also backwards. I didn't really look at that. I got that from Wikipedia, so you got to watch out for things. So basically what's going to happen is our telomerase is going to come in here, and it's going to put in bases here. Or let me think about this. Yeah, it's going to put in bases here, and then it's going to keep on putting bases. It's going to go beyond that. And then we're going to come in with polymerase delta, and we're going to fill in those ends. We're still going to ultimately wind up with that end replication problem. But what we can do is we can extend the telomeres, because every time you duplicate your DNA, you chop off a little bit more, you lose a little bit more, a little bit more. Telomerase goes the opposite direction. It makes them longer. So it keeps your chromosomes length stable sometimes because it's cell dependent. Some of your cells, like cells that you need to have replicate all the time throughout your entire life, have active telomerase. Some of your cells terminally differentiate and they turn off the telomerase so that it acts like a clock. The DNA gets shorter. As the DNA gets shorter, you have somewhere here, you have your coding sequence. And as it gets shorter, you're going to start impinging on the coding sequence. So that acts like a clock in aging. And even if you have a son and you got $5 billion and you want to take your son's blood and inject it in your veins to remain young forever, you're just a very rich idiot. I guess if I were the biologist telling him, I'd go, okay, just pay me $1 billion and I'll tell you, sure, it'll work. No, I wouldn't be that way. Okay. The other thing that happens, so we've solved this problem with telomerase. And before I go on to what happens next, in my own research, I used human foreskin fibroblast cells. In other words, baby was circumcised at the hospital, the tissue was donated for science, and the Shea Wright lab got these tissues from circumcised babies, which was not a problem because UT Southwestern's is part of Parkland Hospital is run by UT Southwestern. You may have heard of Parkland Hospital. That's where JFK was pronounced dead, although he was very dead before he got there. It's the second busiest maternity ward in the world in Dallas, Texas. The second busiest maternity ward in the world is in Dallas, Texas. Yeah, it's in India. But think about this. Think about China. Think about all the countries that have massive megacities. The second busiest maternity ward is in Dallas. Why is that? Why would Dallas have more babies than anybody else? I'll tell you why it is. Because of racism. In the county proper and the surrounding counties, they don't want ICE raids because they would lose their gardeners, their housekeepers, all of those things. But they don't want those people to have health insurance. So all of those people have to rely on Parkland as the only hospital they can have their baby at. That's pure out racism. You may disagree with me, but it is an amazing place to walk to. You go into Parkland and there's like 6 or 7 women coming out in wheelchairs and 6 or 7 coming in constantly. But anyhow, the Shea Wright lab got some of this tissue from Parkland, and they activated the telomerase. They permanently activated the telomerase. They did some recombinant engineering, and they turned telomerase on so that it's permanently active. Why is that? Why would they do that? Well, when you're doing cell culture, cells and culture are not in the same environment as they are in your body. So you're already selecting for cells that are slightly different. You're selecting for cells that can survive in the culture media. You're selecting for cells that do the best in the culture media. The easiest cells to grow are cancer cells. We started our research with HeLa cells. HeLa cells are incredibly aggressive cells. They can contaminate other cultures, seems almost like magically. But the problem is if you're doing your research in cancer cells that have been selected for even more like cell culture, you're very, very, very far away from what real conditions are. And what you see happening in those cells may have no connection to reality in a normal cell. So what Shea Wright did is they took and turned on telomerase and made it permanently active. So these cells, yes, they're still selected for because they grow in the media, but they're the closest thing you can get to cells that are in the body, they aren't cancer cells. They're just human cells, fibroblasts, skin cells that you can use for research, and they give you a model that's closer than other in vitro models. Okay? So I use those cells in my own research. So telomerase, important stuff. But then we have the end replication problem. Yeah, I've got my telomeres longer. Cool. We're going to see how the telomere length is regulated. And we're going to see that here in a second. But here's my 3' overhang. I got my 3' on the bottom on this one. So what happens is we have some proteins that that fold the telomeres over, and they have strand exchange. Now this is all, the telomeres just completely repeats, whatever the sequence is. I showed you one of the sequences that's the normal sequence, but there are variants. So you just have these same sequence repeated over and over and over. That's what the telomere is. There's no coding information here. So yeah, it's really easy to make this loop and have strand exchange happen so that you have this structure form. And since there's strand exchange, that fold stays there. It no longer looks like a double-stranded break. The DNA repair mechanisms are going to ignore it. They're not going to try to glue chromosomes together because they don't look like double-stranded breaks. Does that make sense? All right. So we're doing some hand waving. We're not going to worry about the proteins involved, but you should know about the telomere repeats, the 3' overhang, and the strand exchange to make these T loops at the end of T for telomere. Okay. Now this is what I was alluding to a second ago. This is an experiment that was actually kind of cool. Yes? By the telomerase, yes. So there's inherently some already, but that repeat continues. That's what the telomerase does. It uses the RNA part of the molecule as a template, and it just keeps using that same template over and over and over as it extends. So they're always this about 6 base repeat. Mm-hmm. I'm sorry? Not the cell, the DNA, the chromosomal DNA, yes. That's how they close it off, so that the DNA repair mechanisms don't try to glue chromosomes together. Otherwise, they would look like double-stranded breaks, and you don't want double-stranded breaks, and you have mechanisms in your cell that say, oh, that's a double-stranded break. My job is to fix that. Okay, does that make sense? All right, so let's talk about this experiment really quickly. Doesn't say what I forgot what organism they used for this or what cell line they used for it, but doesn't really matter. They made some artificially long telomeres. They made them longer than normal. And they also made a set of cells that had these short telomeres, shorter than normal, right? And so we start out here. This is increasing telomere length. These are long telomeres. The population of cells has a distribution like this. Over multiple cell divisions, it becomes a normal distribution that matches what the other cells that have normal telomeres are. And if you go the other way and you make short telomeres, you start over here, and this distribution starts spreading this way, and you get the same distribution. This shows us that telomere length isn't just arbitrary, it's regulated. So Shay Wright had to deal with that as well. It's never as easy as, oh, I just flipped the switch and everything did what I wanted. So as I said, telomere length can be used as an aging clock. If you turn off the telomerase, every time the cell gets replicated, the DNA gets a wee bit shorter, wee bit shorter, wee bit shorter. That acts as a clock. If you have a cell's line that is constantly, because the environment or whatever its job is, like making stomach acid or lining the stomach, that's my favorite one to talk about, then you keep the telomerase on, because these cells you just need to be making cell division all the time, and you don't need them to aging out. That makes sense? Okay. All right. I had Google's AI make that, so we can see how AI is getting better. We're talking about DNA repair now. And so I had it make this little cartoon to Gemini. I'm a Gemini, so maybe that's why I picked it. I don't know. I had to make this little cartoon uh, to illustrate what we're going to talk about. We're going to see DNA damage. DNA damage is occurring to your DNA as we speak. It's not some rare event. It's happening constantly. And you have trillions of cells. So once what they're showing here is mutagens or ultraviolet radiation or cosmic rays or X-rays, which would fall under mutagens, I guess, can damage DNA. And we'll talk about some of the ways it can damage DNA. So the first thing we have to do is we have to have proteins that scan the DNA looking for lesions, that is, places where there's damage. And then we have to either excise the damage, this is base excision repair, and then ligate, synthesize new DNA and ligate it, or we have to take a double-stranded break and either do homologous end joining or non-homologous end joining. So we're going to talk about all of these different ways of DNA repair. All right. So I'm not going to talk about coupling. I had it in here, my show, and I decided not to, that I don't want to get too confused about coupling. So I do want you to know that this nucleotide extension repair that we talk about is related, is connected. How do I want to say it? How did I put it here? Coupled. I can't even think of the word. To transcription. That's not exclusively coupled to transcription, but genes that are being transcribed are probably your most important DNA because you're making those proteins all the time. You need those proteins. So this coupling ensures that you efficiently repair your most important DNA is the idea here. All right. So we're going to talk about how we get spontaneous damage. That's the mutagens or the UV radiation. The double helix lends itself to being readily repaired. He who shall not be named because he's a worthless piece of carbon, Crick and his partner Crick saw that immediately when they figured out the structure, that this lent itself to repair and replication. There's more than one repair pathway. It's like, okay, we have this repair pathway. We have several different kinds of damage that occur, several different pathways to fix the damage, depending on what the damage is, where the damage is, and when the damage occurs. We have damage detection, and we have the ability, we've talked about before in your education, the checkpoints in the cell cycle. That if the DNA is damaged, that you can delay the checkpoint, delay the cell cycle until the DNA repair mechanisms have an opportunity to do their job. And if the DNA is so damaged they can't do their job, it typically turns on the apoptotic program to get rid of that cell. Okay. So, what I wanted to show you, I thought this was interesting. I don't expect you to memorize this. But these are inherited syndromes that have defects in DNA repair. The one that I'm most familiar with probably is xeroderma pigmentosum. That's a skin disease, skin cancer, but it's also xeroderma pigmentosum can be induced if you work out in the sun without any sunscreen all your life long. And if you were a kid living in Hawaii and never wore a shirt because you were in Hawaii and you were there for several years, it means you have to get checked for skin cancer all the time. But we have all of these different syndromes caused by defective DNA repair. They're inherited syndromes. And we have more. And you guys are probably most familiar with BRCA1 and BRCA2. And implicated in breast cancer and prostate cancer. Yeah. But again, all sorts of syndromes. These DNA repair is super, super important. Why is it so important? Because your DNA is being attacked all the time. It's being attacked by the very chemicals that are normal chemicals in your cells. You have reactive oxygen species. So you get oxidative damage. It's an aqueous environment. So hydrolytic attacks occur. That's just from its own environment. That's not anything external. That's not a mutagen. That's not UV radiation. You have just the spontaneous DNA damage happening in your cells just because. And how can you damage your DNA? Well, we're going to look at this from hydrolysis. You can have depyrination, depyrimidation, deamination, and methylcytosine deamination. We're going to talk about these two. We are going to talk about deamination too. So I'll say these three. And we're not really going to talk about oxidation, but I want you to look at this. This is the number repaired in a mammalian cell in 24 hours, the number of damage. They calculated that depurination happens 18,000 times in a cell over 24 hours. Think about all your cells. Depyrimidation, a much rarer event. Cytosine deamination, much rarer. Methylcytosine deamination, not that prevalent. The ring-saturated pyrimidines are the most prevalent down here, but this one really sticks out. That's not counting the cosmic rays, the background radiation, and the mutagens you're exposed to. That's just, well, it probably is. I take that because they can't get rid of some of that stuff. All right. Why did I put this one in here? I don't remember why I put that in there, so I'm not going to worry about it. Let's talk about depurination. This is a hydrolytic attack. What are the purines? Guanine and adenine. They're the two-ring nitrogenous bases. So here we have guanine. Here's our DNA strand. Here's our 5' side. Here's our 3' side. And we assume that this keeps on going this way, so 5', 3' down that way. And we have a hydrolytic attack, and it winds up taking off the nitrogenous base. That's what we're talking about happens 18,000 times. Depurination. You lose either adenine or guanine off some of your bases. And that has to be repaired. Now, luckily, that's a really easy repair because it's going to leave the other base. If you had a G, it's going to be a C. If you have an A, it's going to be a T on the other strand. So you can just match and put the right thing in there, and Bob's your uncle. You're all happy. So that's not a major thing. Having 18,000 attacks like that over 24 hours in your cells, really not that big a deal. But this is what we mean when we say depurination. We lost the nitrogenous base. Well, if we can have depurination, we certainly can also have, I thought I had depyrimidation. Okay, well, we can also have depyrimidation. I'm going to go home now. All right. Again, deamination, here we have cytosine, and we have this amine group hanging out here. So this is cytosine. It's a pyrimidine, okay? But we have a hydrolytic attack. We don't take off the entire base. We attack the amine. And what happens when we do that is we turn the cytosine into uracil. Uracil is not a DNA base. It's an RNA base. So you have uracil occurring in your DNA due to this spontaneous damage. Again, a pretty easy one to deal with. Uracil doesn't belong. You cut the uracil out. You put the guanine, cytosine back in. Okay. You're going to have guanine on over here on the other strand, so you know what to put in when you chop that out. All right, now this is what I really want you to know. I want you to know all about these. There's another way that you get damage. I talked about the kid in Hawaii without his shirt on and people who sunbathe all the time. It increases your risk for xeroderma pigmentosum. But what it definitely does is causes more thymine dimers. So thymine dimers happen. Here we have a single strand. Again, this is our 3' end. Here's our 5' end. And we have two thymines next to each other. And we talked about base stacking, and we talked about how those pi electrons are interacting with each other. Well, that's all fine and dandy. But we get a shot of UV radiation. And remember, these are highly conjugated rings. And pi electrons are delocalized electrons, and they absorb UV radiation really, really well. And that can cause these guys to actually make bonds to each other. These are no longer just bound by the sugar phosphate backbone. The thymines themselves dimerize. So you have to deal with thymine dimers. All right. So if you can't repair your DNA, I showed you all that laundry list of inherited conditions that were due to DNA repair problems. If you can't repair your DNA, that means you're going to wind up with mutations. Here we can see we have this deamination. So we have a uracil there. And if we can't get rid of that uracil, when we replicate this guy, what do you match up with uracil? You're not making RNA. How do you know to put an A there if there's a uracil there? It's supposed to be a T if you have a uracil. Uracil doesn't tell you to put anything there. So it's going to cause a mutated sequence. The other strand, because we have semi-conservative replication, the other strand in this case is going to be perfectly fine with its daughter strands. Yeah. But DNA repair, here we have depurination, and like I said, it leaves behind the thymine, and we have a mutated sequence over here. The AT pair has been deleted, or we have the old strand here. Again, this is no repair. So basically what happens in this case, we have depurination and we lose this base. And if that's in the coding region, we've just changed the reading frame. Luckily, there's only about 1.5%, 2% of your DNA is coding DNA, but nonetheless. Okay. So the helix is readily repaired. We talked about base excision repair in DNA replication. But we also have DNA base excision repair mechanisms that are operational all the time. The mut-S, mut-L system is specific to having topoisomerase and replication. There are multiple pathways, multiple systems. This is where I want you, I'm not going to worry about learning all the details of these other systems, but you need to know they exist and have some idea of what happens. All right, so we have this deaminated cysteine that gives us our uracil. There is this protein called uracil DNA glycosylase. So what does it do? Let me, does it show up there? Yeah, this doesn't really show it. Okay. I'm just going to do an abbreviated DNA here. And then we have, what did I say we were doing here? Yeah, uracil. We have our uracil here. I'm just going to put U there. This linkage of carbon number 1 is a glycosidic bond. That's a sugar. That's glyco. So all we do is chop off this guy. Now we don't have any base there. Then an AP endonuclease can come in and repair the missing base. Well, it also gets excised. There's another enzyme that I thought they put that in there. Oh, it's okay. Uracil glycosylase gets rid of the uracil. You have the missing base. Now we can excise that guy. Well, yeah. And so now showing, this is where I was thinking about they showed the proteins. An endonuclease and phosphodiesterase. It's up there. It's got to remove the sugar phosphate. Because that's all we got left. We got the sugar and the phosphate. We don't have the base there anymore. So they nick it. They remove it. And then DNA polymerase can come along and stick in the right guy, and ligase, if needed, can come along and ligate it, and we're Bob's your uncle. We have fixed that lesion. I'm on Bob's your uncle today. Why am I on Bob's your uncle? Okay, so helix lends itself to being readily repaired. This is just that here showing a pyrimidine dimer, a CNT, excision nuclease, just like we saw before in replication. We don't just excise the one base. We take out a chunk of DNA, and then we have the DNA helicase come in here. Remember, this is not during replication. This is just good old times. And then we polymerize and use this strand to replicate. But there's a problem here. Okay. Previously, the uracil was the damaged base, and so nothing ever happened to the G, so I knew that once I got rid of that base, I needed to put a C across from it, a cysteine. How do I know what strand to use here? Should I chop off part of this guy? Well, yeah, I'm going to chop off that. It's a dimer, that's right. I was thinking, but one of the things that we'll see in a minute, this one is a dimer, so ignore me, but when you have other lesions, how do you know which strand to use as the template? And the answer is you don't. There's a chance of getting mutations, depending on what strand you use. We'll see that in a minute. All right. So, detection. Glycosylases can recognize base flips. So what happens, you get the base, it flips out because uracil doesn't really have the right configuration for the hydrogen bonds so with DNA. And it's not across from a adenine, it's across from a guanine. So it doesn't quite fit. So it flips out, and this is what the glycosylase recognizes and chops off that glycosidic bond. All right. I'm going to go really quick here. This is just, we have double-strand break repair mechanisms as well. Here we have an accidental double-strand break. We use a nuclease to chop off, we clean off both ends, clean up both ends. That means it's likely that we lose some nucleotides when we do this. This is non-homologous end joining. So we have these two ends, they're not homologous, we just stick them together, but we trim them a little bit first. So we have a little bit of deletion there. All right, we also have homologous end joining, where, wait a minute, I was looking at this one. Did I get my titles backward here? That's non-homologous. Why is this also non-homologous? I think that's supposed to say homologous there. But in any case, there's our break. We process it, same thing. Oh, here's the homologous recombination. This is part of the repair mechanism here. This stuff I just want you to be aware of. I'm not going to spend a lot of time. We talk about meiotic crossing over, but besides just crossing over, we also get base conversions or gene conversions happening by some of these little things that can happen during replication. Again, it tends to increase our genetic variability. And I was going to talk a little bit about transposons, jumping genes. These are really cool. I'm not going to worry about asking about transposons, jumping genes. We have a lot of viruses do this, and they stick their self into your DNA. Here's a case where the transposase gene includes AMP resistance. And that's a problem because this guy can excise itself and go to another bacteria and insert itself in that bacterium, and now that bacterium has ampicillin resistance. This is one of our things we have to worry about. There we go, Finn. That's it for what's going to be on the next exam. And that last bit of this lecture I went breezing through. That was more an FYI thing. The things I spent time on, you need to know. Any questions, concerns? Nope. All right. Well, then have a merry, merry, happy weekend. Spring break is coming up. Let me turn off this microphone first here and stop the recording, and then I will.